So this will be our first of two presentations where we start to go through some of the cell organelles here. And so we're going to be looking at these different organelles, their functions, what they do in the cell, how they help to maintain homeostatic control. First up is our outermost organelle in the cell, which is the cell membrane or the plasma membrane. The plasma membrane, remember, is made of these phospholipids, these molecules with the polar head and the non-polar tails that have that hydrophobic, hydrophilic interactions. Now the membrane is found in all cell types. So this is found in pro and eukaryotes. It's semi-permeable, meaning that some substances can pass through the membrane and others cannot. And so we have some things like oxygen, CO2, water, right? These substances can actually pass right between the lipids from the outside to the in or the inside to the out of the cell. Other substances have to pass through things like channel proteins. So we talked about these in active transport, both for facilitated fusion and active transport for larger things to move in and out. And those things would include things like glucose, right, and amino acids, AA. And that's how they would enter next to the cell. Also contained then in the membrane, or actually sticking out of the membrane, are these things that are called receptor proteins. So these are accessory proteins that stick up out of the membrane, and their job is to receive signals, okay, from other cells. So receptor proteins. Looking at these receptor proteins, they're tasked basically with picking up signals. So right here, here, and here we have different receptor proteins, and they're picking up some sort of message from somewhere else in the body. It could be an adjacent cell, like over here, okay, or it could be through the blood. And so what are these signals? These are things like hormones, okay? So we're talking about things like estrogen, testosterone, insulin, items that are traveling through different areas of the body. Basically, these receptor proteins have a specific shape that sticks out of them, and the molecule that they're supposed to sense will lock into that shape, just like an enzyme protein interaction. And so that molecule will lock in, and then some signal will be sent into the cell. Okay, the actual molecule might pass into the cell, or a message will reach the nucleus and result in some sort of change. Now, when we look at that membrane, you have to remember it's the bilayer. So we have the inner layer and the outer layer of phospholipids. Remember, they've got the tails that are scared of water and the heads that love water and orient towards water. And these lipids are going to be the most abundant model, these phospholipids. We also then have some other things embedded there. We have the proteins, like the channel proteins that allow items to move in and out, remember? Things like glucose, amino acids, and large ions. We could also have embedded in there some carbohydrates. These are for structural purposes, to make the membrane more rigid. And then we have our receptor protein type molecules that stick up and out of the membrane. The membrane is, though, not solidly attached. So these lipids can move in any direction, and so this is often called the fluid mosaic model, because it's a mix of substances that don't have permanent adhesion or places defined for each other. Next up then is a layer of the cell or an organelle that's found only in eukaryotes. That's going to be the nucleus. This is eukaryotes only. Remember, eukaryotes, those organisms that have membrane-bound organelles, right? And this is going to be the largest organelle in most cells, and its basic job is to store and protect genetic material, okay? And so this is where you'll find all the DNA in a eukaryotic cell will be in the nucleus. It protects it. It has a special pH that it maintains for the genome, and it keeps it's separate from mixing with some of the organelles that might cause it harm. Now, inside then the nucleus, in most eukaryotes, we can also see what's called the nucleolus. This is a darker central region inside the nucleus. And this is actually where your ribosomes are made. So those protein factories made inside the nucleus, and then they pass out through these. You'll notice there's gaps in the nuclear membrane called the nuclear pores that let things move in and out of the nucleus. So the nucleus, like the plasma membrane, has a double layer. So it's got two layers of lipids, and then it's got passageways, the nuclear pores that go through it. That's for things that enter and exit. So it's semi-permeable. It separates the DNA from the rest of the cell, and that's mainly for protection, keeping it from being damaged by environmental factors. And it's in charge of all kinds of different activities. So it controls the metabolic rates, controls the reproduction of the cell, gene expression. So genes are turned off and on inside the nucleus, things for different traits in organisms. So this is a look then at the different DNA structures that might be found inside the nucleus at different times. So this right here is the actual single-stranded DNA. Okay, it's a double helix. So there's two strands, purple and blue there, that are coiled around each other. This is the level at which the models around my room are. Now, DNA is never that loosely packed. It's actually, during normal cell functioning time, it's more packaged up like this. So this structure is called chromatin then. Chromatin is the somewhat wound structure of DNA found during normal cell operations. The idea is that if it was all loose like it's pictured down here, it'd really be too jumbled, too piled up for normal cell activity. So your cell keeps it partially wound. And if it needs to access a gene, it can go in here, it can remove these histones and read whatever genes it needs to find. Over here then, though, is what's called a chromosome. Okay? Chromosomes are those X-shaped structures that you would typically think of when you think of DNA. And this is that chromatin that's been taken and it's wound on itself over and over and over again, and then wrapped again a third time around. And what it's done is it's really packaged that DNA up. Okay? And this purpose here is for division. It's for transport. When the DNA has to be divided up during either mitosis or meiosis, the cell wants it compact and easy to deal with, and so it forms that chromosome structure during division times only. This would be eukaryotes. Prokaryotes have a single chromosome, and it's a circular chromosome. So a prokaryote has only one, and it's a circle, not a strand, and so it's attached. There's no start or no end. This next structure is going to be for pro and eukaryotes, and that's the cytoplasm. The cytoplasm found in all cell types. This is the fluid, pictured here in yellow, that fills the cell. Now that word cyto, remember, means cell, and plasm means fluid. So the word means cell fluid. Now this is a really important but often overlooked cell structure. Its job is really to support and protect the organelles. So the organelles are floating around in the cytoplasm all the time. What that is doing is it's cushioning them, it's protecting them, and then the cytoplasm also is the transport medium in the cell, and so it contains all the organic and inorganic compounds for the cell, things like oxygen, but also things like sugar and amino acids, all right? And it then transports those materials through the cell. It does that through a process called cyclosis, which is when the cytoplasm is actually pushed by the cytoskeleton and circulated through the cell, such that any materials that are on one side can be accessed or transported and are available at the other side of the cell. You'll actually see this in Elodia. You'll be able to see the so in Elodia, they have a square cell structure and they'll have a large vacuole. But what we'll watch the chloroplasts, these little dots. And in Elodia, they'll circulate. You'll watch them as they circle around the cell. That's because the cytoplasm is streaming, called cyclosis. Our next structure then are vacuoles. Vacuoles are storage centers, okay? And so pictured here's a very large vacuole found in a plant cell, okay? And then animal cells, they don't tend to have large vacuoles. They have these smaller structures. Sometimes we call these vesicles. It's a word for small vacuole, okay? And so these can do a lot of things. They're typically storing food, water, and waste, okay? But there are some specialized vacuoles like this vacuole in the paramecium. This is the contractile vacuole. It can pump out excess water, preventing that cell from swelling and bursting due to osmotic movement of water, okay? And then, like I said, plant cells are characterized by large vacuoles. Animal cells, much smaller vacuoles, oftentimes called vesicles. So this slide's just looking at that large central vacuole in a plant. So here it is again. okay, mostly filled with water. And these vacuoles in plants are really important for the structure of the plant. So this slide then is focusing on those large central vacuoles in plant cells, and it's looking at the turgor pressure. Okay, remember turgor pressure is the pressure exerted from the inside of the cell. You can think of it like tire pressure in a car or a bicycle, okay? And it's made by the water. Now, what's happening here is plants like to be in hypotonic solutions, which remember means a high concentration of water on the outside, low solute. And so water moves in, and what that does is that inflates that central vacuole and causes that vacuole to push out against the cell walls, providing structure to the plant. And so when plant cells then lose water, when they're in iso or even hypertonic solutions, they tend to lose water, turgor pressure decreases, the cells tend to kind of shrink in a little bit, the cell walls lack that structure provided by the water, and so the plant will wilt or droop. Moving on, then we're going to go to a structure found in both plant and animal, but not bacterial. So this is eukaryotes only. And that's the mitochondria. Okay. Now everybody always says the mitochondria is the powerhouse of the cell, right? So what does that mean? Okay. The mitochondria's job is to take chemical energy in the form of sugars and turn them into an energy form that the cell can utilize to make things happen, which is ATP. All right. We're going to go through the process of cellular respiration in a later unit that's converting sugar into energy, and we'll look at the formula and all of that then. But for now, we're just going to focus on the structure here. So this is a mitochondria pictured here. It has two main regions that we're concerned about. This inner membrane system that's very folded and convoluted. This is called the cristae, okay? And so what's happening is the reactions for cell respiration are happening back and forth across that membrane. The folds make more space or structure for those. And then we've got the fluid that fills that inner membrane, okay? That's the matrix, the mitochondrial matrix. So this is just showing us a close-up view of the cristae, the membrane, and the matrix, the fluid, all right? And so this is interesting. What I want you to note coming up of importance here is there's two membranes in the mitochondria, and we'll revisit that here in a second. So the interesting thing then, I think the really neat thing about mitochondria is that they have two membranes. Okay? If you look close enough, they actually have their own genetic material. We call that mitochondrial DNA, mitoDNA. Okay? That's passed from, at least in sexually reproducing organisms, from the female parent to the offspring through the egg. It's unique. It's separate from your regular genetic material. And so your mitochondrial DNA is identical to that of your maternal grandmother. So your mom's mom and your mom's mom's mom, you have the exact mitochondrial DNA of those other people. The mitochondria also has its own protein synthesizing enzyme. So what this is telling us, has two membranes, has its own DNA, makes its own enzymes and proteins. And so we've realized is that we think that mitochondria, okay, used to be their own independent cells. Okay, and this is called the theory of endosymbiosis. Endosymbiosis. Right? What this word means, endo means into, and symbiosis is permanent association, okay, or organisms living together. And so what we think happened is mitochondria used to live on their own. At some point, they were engulfed through phagocytosis into an early eukaryotic cell. Once there, that cell did not break them down and instead allowed them to live inside of the cell membrane. That cell protected the mitochondria. The mitochondria, as a result, began producing ATP and giving it to the cell. And we ended up with eukaryotes, right? And another piece of evidence for this is that all animal and plant cells, and so we've got protists, fungi, plants, and animals all share mitochondria, and they all have mitochondrial DNA, etc. And that's the endosymbiotic theory, so that mitochondria used to be a separate organism that has been integrated into modern day cell types.